Hi guys, welcome to another Biology with Miss Cherry. Today you'll be pleased to know we're moving away from plants, away from insects, away from fish, and we're starting to look at humans. So have a go at thinking about these 3 questions while you're waiting to get started, just to get your brain thinking, get your knowledge juices flowing. That's a horrible way to put it, but you get my drift. How do we exchange gases across the membrane? What are the major structures in the lungs? And what is the process of taking in and releases gases from the lungs called? Spend about 3 minutes on these questions and then we'll get started. So this is lesson 3.7. It follows on from lesson 3.5. There was another practical that was meant to be in there, and we're looking at gas exchange and ventilation in humans. So hopefully you said about the breathing in, breathing out process being called ventilation, not breathing. And we're going to start by having a look at the respiratory system. So think on OneNote, I've put this diagram. You should be able to see it and label it up yourself. So pause now and play again in a few minutes when you're ready to go through the answers. So we're going to start up in the top right corner, and we've got the nasal cavity, obviously where you breathe in through your nose, and we've got the oral cavity just below it. You could have said mouth as well. If we carry on down this right hand side, we've got the bronchi, and then we deviate down into the bronchioles. Notice that there's no alveoli on this structure. Carrying on round the bottom corner, so the apex of it, we've got the diaphragm, spelled diaphragm, and we've got the ribs that are there to protect the lungs. The next picture round is actually looking at the little shaded bit behind the lungs, which is the heart, and then we have got the lungs themselves being highlighted in the next box up. And the final couple are a little bit tricky. So we've got the trachea and the supporting structures. So we've got the rings of cartilage that we're going to have a look at over the next few slides. And cartilage is spelled cartilage. And the trachea itself. Don't need to worry too much about the larynx or the pharynx, which you might do in another subject. We're just worrying about the trachea itself and then the cartilaginous rings as well. Now, the first thing we need to think about, like we've done with all of these organisms, is why we actually need a respiratory system. Why do we need an entire body system dedicated to the exchange of gases? And I want you to spend a little bit of time thinking now. I need 3 reasons as to why we need that respiratory system. So pause, give me a few bullet points, and then play again when you're ready. So first thing you might have said is that we need that constant supply of ATP, and the oxygen has to be taken in to produce ATP, that respiratory process. And then that carbon dioxide has to be removed before it becomes harmful. You might also have made some points about a high metabolic rate, and remember that's saying that we need to take in oxygen to produce that energy, that ATP, to then produce the chemical reactions or to have those chemical reactions happening. And unlike any of the other organisms as well, we need to think about why we've got lungs. So why don't we have spiracles? Why don't we have stomata? What is so key about us having actual organs to do this gas exchange as well? And we have lungs inside the body for various reasons, and mainly it's to do with protection and dehydration, like it is in any of these situations. But mostly it's the fact that the air isn't dense enough to support the lungs and to keep them open if they were on the outside of the body. And likewise, the air isn't dense enough to protect the lungs. So if the lungs were outside on the body, like shoulder pads, for example, the air wouldn't be a protective layer. It would just be lungs on our shoulders. Okay, so instead, internally, we can use bones as a protective mechanism, as like a little shield. And finally, if our lungs were on the outside of our body, we'd lose water vapour. So remember, when you breathe out, if you breathe out onto a cold window or a mirror, for example, you get that water vapour forming the little steamy layer. If our lungs were on the outside of our body, they'd become dry and shriveled and they'd lose too much water vapour and we would dehydrate really, really quickly. Now, we're thinking about breathing and ventilation today, and we're going to be using some new terminology that you might not have heard about, although if you do sport, you might have heard about. And we say that lungs are supplied by a tidal stream of air. So I want you to think about what that term means. Think about what a tide is, maybe what a tidal stream is, and play again when you're ready, when you've made a couple of notes about what you think that word means or that term means. Now, a tidal stream is a constant stream of gas towards or away from a set point. Now, in us, that means that we are taking in oxygen and removing carbon dioxide to and away from the lungs, the alveoli, the alveolar walls. And it means that we're replenishing and removing, so we're replenishing oxygen and ensuring that we've got the, not nutrition, but the good stuff that comes with oxygen, and removing that dangerous carbon dioxide waste product. And this is more just a knowing kind of point. It's not really applicable in any other situation. It's more a kind of term that would be used in the stem of a question. So the lungs are supplied by a tidal stream of air. Explain the process of ventilation, and then you would be able to know what tidal stream meant and what is expected of you. Now, the next bits of the lesson are looking at the structure of the lungs, the structure of the respiratory system. So you're going to have to bear with me because we're going to be doing a lot of me talking and you making some notes just for a little bit, and then you guys will be able to get on with some stuff by yourselves. I'm sorry, you could do this independently, but you said last time when you did the cell stuff independently it was a nightmare. So we'll just go through it together. Now, the lungs are a pair of lobed organs. And lobed means that they're made of lots of separate parts that are linked together by tubes. So you can see here that even in this photo of the lungs, there's lots of different parts that make it up. There are a few lobed organs in the body. So we've got the lungs, but also the liver has lobes too. And all of these lungs, or both of these lungs, not all of these lungs, are made up of lots of little tubes inside or internally called bronchioles, and then they branch off into the alveoli. And structurally they are supported and protected by the ribs. So support-wise, it means that we've got some tissues that stick to the side of the ribs where the intercostal muscles are and keep the lungs there. They're not sagging down due to gravity. The trachea is the organ or the tissue that enables gas inhalation and exhalation, and it's the passage from the mouth to the lungs themselves. Now, these are flexible airways, a bit like the tubes on a Hoover, that are supported by rings of cartilage. Okay, and we can say that those cartilage rings are also called D rings because, surprisingly, they look like a D shape. And you can feel them if you put your finger or your thumb on your throat and you just rub your hand up and down. You can feel those D rings of cartilage. And that cartilage is there to prevent tracheal collapse. So if you're breathing in or breathing out, the cartilage is still holding the trachea open. And the trachea has got muscular walls, okay? So it's a contractile tissue that have got both ciliated epithelium and goblet cells. And I want you to think and do some research for a couple of minutes as to why goblet cells and ciliated epithelial cells are really, really important in the trachea and what would happen if they were absent. So pause, I want you to spend 5 minutes on this, finding this answer out, and then play again when you're ready. Okay, folks. So the goblet cells are there to produce mucus. You know, things like phlegm that you get when you've got a cold or a chest infection. And the epithelial cells are those cells that have got lots of little finger bits on them that are able to waft. Okay, so if you imagine both of these in the trachea, the goblet cells produce the mucus and the ciliated epithelial cells move the mucus up back to your mouth where you can swallow it again. Now, the idea of those two working in conjunction is that the mucus produced by the goblet cells traps infections. Okay, so it traps pathogens and stops infections moving to your lungs. And the ciliated epithelial cells will move the mucus with the pathogens up to your mouth, and then you swallow the phlegm, disgusting I know, into your stomach where the stomach acid will digest them. Now, obviously, if those are absent, you will end up with a lot more chest infections. There's no protective mechanism to stop infections getting into your lungs. And if we're thinking back to the end of topic 2, this would be an example of a physical defence. Okay, these are non-selective, these are unspecialised, they're always going to work against infections of the respiratory nature. So if we carry on down, so we've looked at the structure of the lungs, we've looked at the trachea. Now we're thinking about what comes next. So the trachea is going to deviate or split into the left and right bronchi, which are very similarly structured to the trachea itself. Now the bronchi are cartilaginous, so they've got cartilage rings, they've got mucus producing goblet cells, and they have also got ciliated cells again. But as the bronchi get smaller and smaller, going towards bronchiole size, the amount of cartilage in their rings also gets smaller, so they become less rigid and a little bit more flexible. And finally then we get to the bronchioles and the alveoli. And the bronchioles are around 1 millimetre wide, and they're only made of smooth muscle. We don't have any cartilage now. And in the process of ventilation in breathing, it's the contraction of this smooth muscle that allows the narrowing and the expansion of the bronchioles, and that depends on the airflow. So if there's lots of air available, if you're breathing really deeply, then the bronchi are going to expand and then recoil. They're going to elasticate themselves back nice and tightly to force that air out. Again, with these bronchioles, they've got another adaptation that we haven't yet seen, and that's that the walls are quite thin. And in the bronchioles, we can start to exchange some gases. This is only a very, very minor fact about the bronchioles. We're focusing more about gas exchange in the alveoli, but it's an important point to remember that they're still very, very thin. And finally, we get to the GCSE favourite, the alveoli. So these are very small. We're talking like 300 micrometers big. And remember, micro is times 10 to the minus 6 meters in diameter. And this is where we say we've got the most gas exchange occurring. Got a thin layer of epithelial cells, so they're all laid in like a brickwork, so laid like a wall. And they're supported by some collagen, so some kind of stretchy material, and also elastic fibres. And it's those elastic fibres, a bit like the muscles in the bronchi, that enable a recoil movement. So if you think about breathing in, you breathe in and expand your chest, and then almost automatically, unless you hold your air in your chest, the recoil of the alveoli is going to force that air out. So even as I took a big breath in there, and I'm speaking to you guys, the air is leaving my lungs, but I'm not forcing it out. And these alveoli are super duper specialised for gas exchange. And I want you to make some notes now of the 3 things that you can remember from GCSE as to how the alveoli are adapted for that gas exchange. So I've done a lot of talking. It's now time for you guys to do some stuff. How do the cells lining the trachea and the bronchi prevent the alveoli from damage? It's 3 marks. You're talking about the cells specifically and how they prevent damage. So pause for about 4 minutes or so. Maybe set yourself a little timer and then play again when you're ready to go through the answers. Okay. Hopefully you've spent some time now having a go at this question, or at least making a little mind map about your ideas. And the first thing that you should have said is that goblet cells produce mucus, which is there to trap dirt and prevent bacteria or pathogens from the air being breathed in. The cilia on the cells are going to move any debris up the trachea and swallow it again into the stomach. And it's this dirt or bacteria that could cause the infections in the alveoli. Now we're going to have a look at the process of ventilation. And it's a different process to what you might think it is. There's actually a lot going on when you breathe in and out, even passively. So let's think about pressure to begin with. Which of those drawings, left or right, shows the particles under a higher pressure, and how do you know? So I want you to talk to me about particles and the space. The particles in the small container on the right have a lower volume, and therefore we've got the same number of particles in a smaller space, which means that this right-hand picture is at a higher pressure. Okay, so remember, pressure is to do with the amount of particles in a given space. And like humans, like everyone, like everything, including particles, we want to move from an area of where there's high pressure, so it's really tightly packed, to low pressure. If you think about it like being on the tube or a really busy train, if everyone's crammed together, you really want to get out of there. And that's exactly the same as what gas particles want to do in the lungs. They want to move from high pressure to low pressure. And this high pressure to low pressure movement, the gradient, is the basis behind ventilation. And ventilation is the word that we're going to call breathing from now, and it's the process of moving air in and out of the lungs. So a way to remember it is a ventilator is like an artificial breathing apparatus. You know, when people get put on ventilators in hospital, it's doing the breathing for them. Ventilation is the process. Before we actually think about the process though, we've got to be aware that there's lots of different muscles that are involved in breathing. So we know about the diaphragm, that's that flap of tissue that's normally quite dome-shaped that separates the abdomen from the thorax. And the thorax is the sciencey bit for your chest or where your lungs are found, and the abdomen is where your digestive organs are, sort of near where you'd say your tummy is. We've then got the internal intercostal muscles, which are sort of between the ribs, so you can see on the diagram here. If you imagine like a rack of ribs, if you have some ribs from like the Chinese or a barbecue place, that's what you're eating, the intercostal muscles. And those things are going to be causing expiration when they contract. And conversely, the external intercostal muscles, also delicious if they're smothered in things like barbecue sauce, if they contract, then you're going to get inhalation. Now, obviously, it would be nice if external intercostal muscles caused expiration and internal caused inhalation. It'd just make it easier to remember, but that's not the case. Okay, so maybe make a note of remembering that internal expires and external inspires. You know, opposites. Makes it a little bit more difficult. I don't know. It's up to you if you choose to use that. Now, when considering ventilation, obviously we need to be able to breathe in before we can breathe out. So we'll start here. Breathing in is also called inhalation and sometimes inspiration. So you're inspiring. Spirometry is to do with measuring the breathing rate. Either way, it's an active process. And we remember that active means that it requires energy. And it requires energy for a number of reasons, but mainly it's to do with the fact that there's lots of muscular contraction. And that is as follows. So the first thing we're going to get is the diaphragm contracting. And what that means is it's going to flatten from a dome shape into a nice, tightly stretched sheet. But what it does is it helps expand the volume of the thorax, of that chest cavity. At the same time, we are contracting our external intercostal muscles, which cause the ribs to move up and out. And you can feel that if you breathe in and take a deep breath in now, you should be able to feel that your chest goes both upwards, so actually lifts away from the floor, and outwards ever so slightly. And in doing both of these, we are going to increase the size in our thorax. Okay, the volume of our chest cavity is going to increase. And at the same time, that's going to reduce the pressure of the organs and all of the cells and tissues and particles inside our chest. And that reduced pressure is compared to the atmosphere, or compared to atmospheric pressure. And that sets up a pressure gradient between the atmospheric air and the pressure inside the thorax. And as a result, the air moves from the atmosphere into the lungs quite quickly down that gradient to equalise the pressure. And that's how you breathe in. So maybe take a second now, pause, maybe put your hands on your lungs and think about that process. The breathing in, as an active process, think about the muscles that are contracting, think about what's happening to the pressures, and then we'll move on to exhalation next. Now, exhalation or expiration is passive. It doesn't require energy, and it focuses a lot more on relaxing of those contractile muscles. So the first thing we're going to have is the relaxation of the diaphragm. It's going to round up again. It moves up, it becomes domed. And that's the first stage of reducing the cavity size. I said that it was passive, and it's true, but there is still some slight muscle contraction. So the internal intercostal muscles are going to contract to pull the ribs together. So if you remember on the diagram, they're between the rib bones themselves. And if those muscles contract, then they're going to move the ribs tighter together and cause the rib cage to move both in and downwards. And that results in a decreased size of the thorax. And if we've got a lower volume, then we're going to have an increased pressure of all of our particles and organs and things like that in the chest cavity compared to the atmosphere. Okay, remember that's what we're doing. We're always looking for a gradient, and as a result, the air is pushed out of the lungs, a bit like if you were to squeeze a balloon rather than just let it deflate. And that ultimately is going to result in an equalised pressure again. When we're thinking about the muscular involvement in ventilation, we've got to be highly aware that the intercostal muscles are only really used during strenuous exercise, so during phases of deep breath as opposed to shallow rest breath. So under normal conditions, it's the elastic recoil of muscles in the things like the bronchi and the bronchioles that enable the air to be forced in and out. So even if you think about breathing now, you're probably not breathing in or out as much as you possibly can. Conversely, if you're doing exercise, maybe you've run for the bus, or in some cases walking up the stairs might be enough, that strenuous exercise, your intercostal muscles are going to be really contracting in order to maximise the amount of air drawn in and drawn out. Now we're going to spend some time doing a little pit stop. It's a 5-mark question. Compare and contrast the processes of inspiration and expiration. So what's the differences that occur when you breathe in and breathe out, and how are they both the same as well? So it's 5 marks. I'd like you to pause and spend about 5 to 7 minutes answering this question. You can do it as prose, you can do it as bullet points, but notice that compare and contrast means that you have to say, if inhalation does this, exhalation does this, if it's a different, or both processes do this. So you have to mention both of them as a single point. Okay, so pause and then play again in about 7 minutes when you're ready. So the first marks I'm going to show you are for the inhalation and exhalation similarities. So they're both using the diaphragm and the intercostal muscles to move gases, and they both require changes in pressure and volume of the thorax to cause the changes to gas movement. Differences, however, you can talk about the diaphragm. So you would say in inspiration the diaphragm contracts, whereas in expiration the diaphragm relaxes. You could have mentioned about the intercostal muscles being involved. So the external intercostal muscles for inspiration and internal for expiration. You could say about the volume of the thorax, and I'll put that down there, of thorax. And how when you breathe in or inspire, the volume of the thorax is going to increase. You're expanding your chest cavity, and in expiration it's going to decrease. And obviously we can talk about the movement of gases in for inspiration and out for expiration, and the changes in pressure that cause those. So the pressure of the thorax is going to decrease when you inspire, and that's why the gases are able to move in. And conversely, the pressure of the thorax is going to increase for expiration, and therefore air is forced out. So each of those lines is a separate marking point, but you had to have both a mention about what happens in inspiration or inhalation and exhalation for the single mark. So there's a maximum of 2 for the similarities and then a maximum of 3 for the differences. If you've got 5 differences, you can still only get a maximum of 3 points. Now, as you well know, quite a lot of your A-level is maths. About 15 to 25% of some papers can be maths. Okay, and this is one of the calculations involved in breathing. And the pulmonary ventilation rate, or PVR, is the total volume of air that is moved into the lungs during 1 minute. And it's calculated with that blue equation. So PVR in dm cubed per minute, which can also be written as dm cubed per min, is calculated by multiplying the tidal volume in dm cubed, which remember we've spoken about at the beginning of the lesson, and breathing rate in breaths per minute. Okay, and that can be written again as breaths to the -1. Okay, so can we think about what tidal volume and breathing rate mean scientifically? Hopefully you can, because we've done tidal volume, and breathing rate is quite self-explanatory. And they're here. So the tidal volume is the volume of air taken in at each breath when the body is at rest. So not under stressful situations, not under any kind of exercise or physiological stresses in any other form. And the breathing rate is obviously then the number of breaths taken in 1 minute, because you can see it's measured in breaths per minute. Surprising that. Now have a go at using that equation. An average breath takes about 4.25 seconds to complete. What is the breathing rate for the person? And then if the average person's breath is 1.2 litres, what is their PVR? So pause, have a go at those. There's a little extension question at the end if you want to challenge yourself, or if you're targeting an A star, especially for paper 3. So wider knowledge is often times the most important thing in a really high scoring essay. So if you did want to have a look at the piece of equipment that's used to measure tidal volume, go for it. So for the breathing rate of the person, we know that there's 60 seconds in a minute. And if each breath takes 4.25 seconds, then we're going to do 60 divided by 4.25. And using a calculator, that works out as 14.12 breaths per minute. Okay, remember we need those units. And then for the pulmonary ventilation rate, PVR, we know we're doing breathing rate times the tidal volume. Breathing rate is 14.12 times 1.2, and therefore the pulmonary ventilation rate of the person is 16.944, or, to 3 significant figures, 16.9 decimeters cubed per minute. Again, don't forget the units. Now, could you have a go, please, at the summary questions on page 144 to 145? I've put a little photo here for you to use if you haven't got the book, although you all should have. So hopefully this will be fine. And if you're finished, have a go at that pulmonary ventilation purple box as well. So we're going to spend the last few bits of the lesson looking at the alveoli and gas exchange, and also then the difference between correlation and causation. So the alveoli, we know, are tiny air sacs in the lungs, very small, and they've got these 3 adaptations for gas exchange. So they've got 1 cell thick walls, they're partially permeable, and they've got a large surface area. But I want you to pause now and tell me why each of those is useful for gas exchange. So pause and play again when you're done. Now hopefully this was quite quick, quite easy. We're all talking about diffusion distance really. So having walls of 1 cell thick, we're going to reduce the diffusion distance. If we're partially permeable, we're going to allow substances through, and again reduce the distance that particles need to travel through to move from space to space. And finally, the large surface area, we're going to increase the space available for diffusion. Okay, and that's just going to help increase the rate of diffusion again. Now, if you remember when we were talking about the gills and things like that, we were talking about countercurrent flow. Now, the process of ventilation, the breathing in and breathing out, is very similar to the process of water flowing over those gills. So the air coming in and reaching the alveolar membrane and then being breathed out again ensures that the medium, or the air, is constantly flowing over the exchange surface, that alveolar membrane. Okay, that means that we're going to get a constant concentration gradient, because diffusion alone, if we would just hold the air in our lungs, wouldn't be enough for gas exchange. We've got to have that constant medium that's transporting the substances away and to the site. So annoyingly, there's lots of abbreviations with this respiratory stuff. So the first one we had was PVR, and now we've got the FEV, or the forced expiratory volume. And this is the maximum volume of air that you can breathe out in 1 second. And you can find this out normally at the doctors, especially if you're asthmatic. They give you that tube to breathe out into, where they tell you to take a deep breath in and then force all the air out of your lungs. Now, diseases that cause constriction of the airways or narrowing of the airways, things like asthma or pulmonary fibrosis, can restrict the amount of air that you're going to be able to breathe out in 1 second. And I want you to think about what impact can that have on a person, both physiologically. Maybe you want to think about growth and development, things like that. Okay, so pause, maybe make some notes, and then play again when you're ready. So here's another way that we can show how respiratory conditions affect the forced expiratory volume, okay, the FEV. Have a look and see if you can see what happens to the FEV in patients with obstructive lung conditions. So remember that the FEV is the volume of air exhaled in 1 second. You'll notice as well that this graph is a little bit of a mess because there's also the FVC, the forced vital capacity, which is the maximum amount of air that you can breathe out after the deepest possible inhale. So that's when you're trying to breathe in as deeply and out as deeply again without a time limit. So what does the graph show about the FEV in patients with obstructive lung conditions compared to the normal person? Pause and play again when you're ready to listen to me waffle through the answer. So the first thing we can see is that the FEV for both asthma and COPD are significantly lower than the normal FEV. Okay, so normal FEV I'm going to highlight in red for you. And that's probably because the constriction of the breathing tubes, the trachea, the bronchi, the bronchioles, is going to mean that there's much less space for air to be forced out of. And COPD is even more significantly reduced compared to normal than asthma. And COPD is caused by a long damage or a long-term damage to the lungs. It's called chronic obstructive pulmonary disorder, and it's where you do things like smoking and you start damaging tissues rather than just constricting them. So maybe you might want to do some research now as to why COPD has a lower FEV than asthma. Now, when we were looking at that graph, there was a lot of inferences made from that on my part. So I said that asthma and COPD cause a narrowing of the trachea, the bronchi, the bronchioles, and that shows a causative relationship, so that the narrowing of the vessels causes a reduced efflux or removal or loss of air from the lungs. But that's not to say that there's a correlation between them. Okay, and it's really important that we know the difference between those two terms. So correlation is where there is a relationship between two things. Okay, whereas causation is when something causes another thing to happen. So there's a correlation between my age and how grumpy I am, but that's not causative. Okay, a causative factor would be like the more miles I drive, the lower the fuel is in my tank. Okay, there's a direct impact of the amount that I'm driving versus how much fuel I've used up and the resulting residue that's left in the tank. So have a look at this graph. It's a really abstract example. It's more just to look at the relationship between the two variables. So what does the data suggest is happening? We've got a number of active Facebook users in black and the yield on 10-year Greek government bonds in the yellow. So the data actually suggests that over time, both the number of Facebook users and earnings on the Greek government bonds is increasing. So they're both going up at a similar rate for the last few years. There is no link between the two. Okay, the Facebook users does not directly affect the government bonds, but because they show a similar relationship, we can say that there's a correlation between them. There's some kind of linkage in the patterns that they show. A causation or a causative relationship can't be identified from a graph alone. And the only thing we can use graphs for is to identify a link or a correlation between factors. However, to find a causal relationship or a cause and effect or a causative method of transmission or development of a disease, we have to carry out an in-depth scientific study. There has to be a lot more background to say that X directly causes Y. Now, obviously, we recognise a graph like on the left-hand side to be true now, so that the number of cigarettes smoked directly influences the deaths from cancer or increases your chances of cancer. But that is based on many years of rigorous scientific investigation. That wasn't just plucked out of thin air and hoped for the best. So now it's your turn. Have a go at the How Science Works, Correlations and Causal Relationships and the Risk Factors for Lung Disease on page 148 to 149. And the questions carry on to page 150. and then use the marks at the back of the book to self-assess yourself. And finally, as per usual, we're going to finish off with some exam questions just to round off the lesson. Breathing out as hard as you can is called forced expiration. Describe and explain the mechanism that causes forced expiration. It's 4 marks. You should have at least 4 sentences. Pause, give it 5 minutes to answer, and then play again when you're ready. The forced expiratory volume, or FEV, is the volume of air a person can breathe out in 1 second. Using data from the first second of forced expiration, calculate the percentage decrease in the FEV for Group B compared with Group A. So the first second and the percentage decrease of B versus A. And your answer is in percent. It is a 2-mark question, I believe. So have a go. The graph here is a little bit bigger, so you can see the values a little bit more clearly. And the final question, the people in Group B were recovering from an asthma attack. Explain how an asthma attack caused the drop in the mean FEV shown in the figure above. And you can go back, you can rewind the video if you want to, or look at the PowerPoint on OneNote to see the video, to see the graph in a little bit more detail. So again, this is 4 marks. Pause and play again when you're ready. So the first question about forced expiration, your marks are here. So you should have said contraction of the internal intercostal muscles, the diaphragm relaxes, or the external intercostal muscles relaxes if you fancied it. The decrease in the volume of the chest or the thorax, and air is pushed down the pressure gradient out of the lungs. For question 2, you should have got 19%. And if you're not sure how to work it out, please let me know and I will go through it with you on the videos. And then finally, talking about asthma, you should have said that the walls of the bronchi or the bronchioles are contracted. You might well know that asthmatics secrete more mucus as well. And the diameter of the airways is ultimately going to be reduced, resulting in the flow of air being restricted or reduced as well. So I hope you found this useful. Let me know if you've got any questions, and I'll see you again next time. Bye!